Okay, so we're going to keep talking about the central dogma. The last video that you saw was the first part of the central dogma, where DNA is transcribed into messenger RNA. Now we're going to focus on that last part, the process of taking mRNA and turning it into a protein, which has a certain function in whatever organism this is being done in. And this process is called translation. So again, we're taking messenger RNA that was created during the process of transcription and translating it into a protein. So we're taking RNA language and making something functional from it. And in this case, it's some sort of protein. So all cells, regardless of if they're prokaryotic or eukaryotic, have some similar parts of this process. And there's a common feature to all cells, common feature to all living things, as a matter of fact. The thing that does the translating, what actually takes mRNA and turns it into a protein, in that complex is called a ribosome. Ribosomes are large complexes of a variety of macromolecules working together. Ribosomes have a large subunit and a small subunit, and they form around mRNA and help the process of translation. So again, ribosomes are present in every living cell type, regardless of if it's a prokaryote or a eukaryote. If you're dealing with a prokaryote, like a bacteria, all of the ribosomes are free-floating in the cytoplasm. If you're dealing with a eukaryote, you can have ribosomes present either in the cytoplasm or in the rough endoplasmic reticulum. So there's one difference here, but remember that the commonality is ribosomes are present in any living cell type. Another slight variation in prokaryotes is the fact that since everything is happening in the cytoplasm, all of the processes of the central dogma, transcription and translation, can happen together, can happen in concert, as a matter of fact. So transcription and translation happen simultaneously in prokaryotes. So we have a diagram here demonstrating that, and we also have a picture. This is an electron micrograph of an actual process of transcription and translation occurring simultaneously. You have DNA here. You have an RNA polymerase creating an mRNA strand, and then you have ribosomes attaching and creating protein. So everything is happening in concert. In eukaryotes, on the other hand, there is a division. Remember that transcription in eukaryotes only happens in the nucleus, and it has to be processed. It has to be capped, tailed, and spliced. And then the mRNA can go out into the cytoplasm, and it can either be translated with free ribosomes in the cytoplasm or taken to the rough endoplasmic reticulum, which is budded with lots of ribosomes. So those are the two major sites of protein synthesis in a eukaryote. As we go through translation, you again need to keep track of a lot of these different terms. So these terms here on this slide are specific to translation. Translation only involves these things. Do not get them confused with other processes we've talked about. Ribosomes only come into play in translation. mRNA, which is produced in transcription, is used in translation to make proteins. tRNA, or transfer RNA, is a part of translation, and amino acids are now coming into play as part of translation. Unlike transcription or DNA replication, the code is not read one letter at a time, an A, a T, a C, or a G. In translation, 3 letters at a time are read. And these are called codons. Codons are grouped in groups of 3. So combinations of A's, C's, G's, and U's, since it's RNA, are read and then translated into specific amino acids. Codons are only found in mRNA. DNA does not have codons. Do not confuse these terms. mRNA is the only time codons come into play in this process. So again, remember back to Module 1 where we talked about building blocks of macromolecules. Proteins are a major macromolecule. And the building blocks of proteins, the monomers that make proteins, are amino acids. And there are 20 amino acids listed here in this table. And the way that we can translate from an mRNA language to a protein language through amino acids is through what we call the genetic code. The genetic code is in the first part of this chapter that you've been reading. But here's this table again. So we only have 20 amino acids, but we have 64 different codons. Remember, this is a codon now. You have to read 3 letters at once. So a given codon codes for a particular type of amino acid. Only one codon codes to an amino acid, but an amino acid can have many different codons that code for it. Take example, phenylalanine has 2 different codons, but UUU can only be phenylalanine. Similarly for serine, SER, UCU can only be serine, but serine could be any of these 4. So we call this redundant. We call the genetic code redundant because you can have only 20 amino acids, but 64 different combinations of codons that can make up those 20 amino acids. And this is our key. This unlocks the protein message, the series of amino acids that are laid down in order to make a protein functional. Transfer RNA bridges the gap. It brings in the amino acids. So you can see these are 3 different representations of the same thing. These are all transfer RNAs. At the top, an amino acid attaches. Only one amino acid can attach to a specific type of tRNA. tRNAs on the bottom have what's called an anticodon, which complementary base pairs with mRNA produced. So again, one amino acid can be brought in by its very specific tRNA, which has a specific anticodon that binds to the mRNA. And we're going to see this carried out in the next few slides. So take a minute and think about all of these different things, and then we'll progress through. Again, translation is a polymerization process, and it has the phases of initiation, elongation, and termination. And you're going to need to know what is specific to each of these stages, specifically related to translation. Initiation and translation involves a start codon. The only start codon that exists is called AUG. Adenine, uracil, and guanine. Remember, we are working with mRNA, so U is present. AUG only codes for one amino acid, and that amino acid is methionine. Methionine is coded for the start codon AUG. So if we look at this tRNA, this sort of blue-green structure here, methionine is attached to the top. The anticodon, UAC, codes for AUG because it complementary base pairs. So that tRNA can only fit on the mRNA when AUG is present in that order. Those bond together, and again, remember ribosomes have 2 parts. So the mRNA lays on the small subunit, and once the start codon has been attached by methionine tRNA, then the large subunit lands on top. So again, methionine, tRNA, AUG. That is initiation. Elongation is the process of the ribosome moving down the mRNA molecule, bumping off a tRNA, bringing a new one in, and then creating a chain of amino acids. And remember, amino acids are building blocks of proteins, and they have very specific bonds. The type of bond that forms in amino acids in a protein are called peptide bonds. Peptide bonds are covalent bonds between amino acids. So elongation is simply the ribosome moving down a specific mRNA strand, producing a chain of amino acids bound together in a polypeptide. Another name for a polypeptide is a protein. And this occurs over and over and over again. So what this is demonstrating is a cycle of tRNA bonding, the formation of a longer chain of polypeptide, the empty tRNA being bumped off the ribosome, and the process continuing over and over again. So termination, which kind of makes sense, ends with a stop codon. Initiation starts with a start codon, termination ends with a stop codon. Termination can occur with one of three types of stop codon: a UAG, UAA, or UGA. When a stop codon is reached, a tRNA doesn't bind, a release factor binds. So you have this string of amino acids here, a polypeptide, attached to the current tRNA. The tRNA is bumped off, and instead of a new tRNA, a release factor moves into this space of the ribosome, and the polypeptide floats away into the cytoplasm. And as a result, the ribosome breaks apart and the mRNA goes away. That mRNA that was used was translated by a ribosome and turned into a string of amino acids, or a polypeptide. Earlier in the semester, we talked about the different types of protein structure. What is produced directly from a ribosome is called the primary structure of a protein. The primary structure is all the amino acids peptide bonded together in a linear fashion. Because of all the functional groups that hang off of amino acids, some are acid, some are base, some are hydrophilic, hydrophobic, they have all of these properties, if you'll remember back to Module 1. As a result, this linear protein can wrap around on itself and form a secondary structure. It could be a very basic helix or a sheet, and then this primary structure folds in on itself once and secondarily folds in on itself to form a three-dimensional structure called a tertiary structure. A quaternary structure is where you get multiple amino acid tertiary chains bound together in a functional group. So again, whatever is produced from translation is the primary structure, and then once it's released into the cytoplasm, it can fold in on itself and form a functional complex. This final slide here is a summary of the process of transcription and translation. So this is the entire central dogma, but it's a very particular type of cell. And there are several key features here that should give away which type of cell it is. But the same mechanics work regardless of cell type. Transcription has to happen. DNA has to be turned into mRNA. Transcription. Once we move into translation, now we're dealing with codons. We're dealing with three-letter combinations that are read by a ribosome. A start codon, methionine, initiates the process. Elongation occurs, creates a polypeptide chain, and a stop codon allows the polypeptide to be released, fold in on itself, and form something functional. So take a minute, review this summary, and again, as I've said for everything in this unit so far, draw it out. Make sure that you know what process you're dealing with. Make sure you know the proper terms. A lot of these terms are being thrown out there, but you need to make sure that you can equate the proper term with the proper process.